Welcome, ladies and gentlemen, to this unique webcast that is a joint production, let's call it, of the Albert Einstein Institute Hospital in São Paulo, Brazil, and the Technion in Haifa, Israel. My name is Rafi Naveh, and I'm a senior researcher at the Sam Nieman Institute at the Technion in Haifa. And I would like to share with you a few guidelines. Basically, this webcast is conveyed as a webinar. Namely, all participants cannot use their microphones or their cameras, only the presenters, which are defined as panelists. Each presenter will have 5 minutes, okay, and we'll try to be very sharp that they will fit within the time. And there is not going to be additional time for questions and answers, but you can use the Q&A or chat facility in the Zoom application and write questions. Now, if you write a question, make sure you indicate to whom the question is presented, okay? So that at the end of the call, we can forward the questions that are intended to a specific speaker to him or in order to respond to the question, okay? And we are going to get back to you with the answers or comments based on what the presenters are going to say. And before we start, I just want to show you the agenda. I'm not going to walk you through that for the sake of time. Within a minute, the president of the Albert Einstein Hospital, Dr. Kleiner, is going to address you with his opening remarks. After that, Professor Landborf, the Technion Vice President, is going to give his introduction, and then there are going to be 12 researchers or leaders that are going to present. We're alternating one from the Technion and one from Brazil. And again, we are going to try to make sure that they all fit within the 5 minutes that we allotted to them. So within an hour and 15 minutes, I hope we're going to get done. So this basically shows the sequence, but we're going to introduce every person as we switch from one presenter to the next. So the first presenter then, or the first person to address you, is the president of the Albert Einstein Institute Hospital, Dr. Sidney Kleiner. Is he available, please?
Yes, I'm here. Thank you, Rafi.
So I'll stop sharing my screen and okay, very good. Here you are.
So thank you very much for this opportunity again to be, I think last month we were together with Alon Wolf. Hi, Alon. And the objective that time was a little bit different, that the friends of Technion in Brazil, led by my friends Salomon Yospi and Salo, they are here. And forgive me if I forget someone in this meeting, but Albert Einstein started to split the division of research in our institution that completed in 2018 20 years of teaching and research. Is a little bit young institution. We complete last month 65 years of our organization, founded by the Jewish community after the immigration after the war here to Brazil. And the hospital was a way to be thankful for the Brazilian society of this reception of the Jewish community after the war. And they imagine, our visionaries and founders, they imagine how to be thankful for this reception to the Brazilian society, giving health to the major society of Brazil. And that's a purpose of the Albert Einstein Society. And we are always supported by 4 precepts, Hebrew precepts that are mitzvah, refuah, chinuch, tzedakah. The tzedakah in name of the social justice, And one of the three pillars, the first one is the healthcare excellence on assistance, the second one, teaching and education, and the third one, research and innovation. And through this third pillar, in terms of research and innovation, that we split it a few years ago when Claudio Terra came from to the institution to start this directory of innovation that 2 years ago we achieved the conception of our first incubator in healthcare in Brazil. That the name, not a coincidence, it's Eretz.Bio. It's the name of our incubator. And through this incubator, we had in the beginning of the pandemics, about 34, 35 startups incubated, all of them in terms of healthcare. And during the pandemics, we reached, and Claudio Terra may be a little bit, he has more details, but we reached 50 startups with all the ideas. Some of them will be pitching here about the project and how we are in terms of innovation. So I'm very proud to put Einstein in this world scenario. We have some partnership together with another institution in the United States and also in Europe. But for me is something that I am more proud to be together and exchanging experience and ideas and innovation with Technion, a very, very respectable organization that I was last year with you in Israel. And I'm very proud to put Einstein in this conversation and to share what we have done in terms of pandemic. So once again, Alon, it's my pleasure to be with you. And I think you can do more and more, much more together in the future. Thank you very much.
Thank you. Alon, please enable your microphone and address us.
Thank you, Tini. Thank you, Rafi. Thank you, everyone, for joining us. And it's very exciting to be here again together, even if only virtually. I promise the first options we have to meet also again as we used to. I'll pay you a visit as we were about to do and we scheduled. And unfortunately, the corona shuffled all the cards. But it's really it's an exciting collaborations that we are establishing between the Technion and Albert Einstein Institute, the hospital. The Technion, opened its door cornerstone was laid more than 100 years ago, but the first class was in 1924. So we are almost celebrating the 100 year of teaching. And from day one, this institution was very special in the sense that the acceptance of students to the programs were not not influenced by religion, race, and gender. In the first class of 1924, there was one student lady who graduated, which was very exceptional at that time, even in Europe and in America, was a class of 17 students in civil engineering and architecture. And since then, Technion was in every junction and every point in development of the country, the state of Israel. I don't think that there are many universities that can actually claim that its graduated, its alumni actually build a country from scratch, from nothing. Most of many of our alumni, as maybe Shlomo would talk about this with his famous book, open startups and are responsible for, I think, 80 or 90% of the successful startup in Israel. And Israel is very strong in this. As everyone else, unfortunately in Brazil, also here now in Israel, we were affected by the coronavirus and we had to go teaching online. But what was immediately noticed is that many of the researchers here took their years of experience in science and converted this into applied science in developing technologies to fight the virus. One of the first things we did is to join with Rambam Medical Center, which is the biggest or the largest hospital in the northern part of Israel, and with the other hospitals in Haifa and the region associated with our medical school, to develop projects. So we had more than 60 labs working on technologies and in different aspects of the pandemic. Some of the technologies are related to personal protective equipment. At least 3 of them already used worldwide: the sticker, the Maya sticker, and the visor with the airflow, and others used worldwide now in America and Europe, and are authorized to be used already. We also were heavily involved in service robotics to help in those corona departments, and those robots are operational now. In diagnostic and in therapeutic, some of those technology you're going to hear from the researchers and professors that are here in this webinar, fantastic webinar. I'm also very excited, as Ines mentioned, is about the collaboration, the very strong collaborations we established. We already established a research collaboration of joint project between the hospital, the Albert Einstein Excellent Hospital, and the Technion. And I'll just give you a head start as just a hint, as our Prime Minister used to do before going online now, that we are now in a very advanced process of establishing key strategic collaboration with the Albert Einstein Institute and Hospital in education even aspects. Hopefully this will bear fruit often. So we are very excited, and we want to thank you really from the bottom of our heart for this warm collaborations that we have between Brazil and Albert Einstein Hospital and the Technion. And I wish you all to enjoy this fantastic webinar and these fantastic speakers. Thank you very much. Enjoy the evening.
Thank you. We're going to turn to the first speaker, who is Eran Friedler. And I already shared the screen. Eran, can you see it?
Yeah, thank you. And
please go ahead.
Thank you, and I'm honored to be part of the presenters in this wonderful webinar. Hopefully it will end up in scientific and also practical collaboration between our countries. I'm going to talk about smart wastewater-based surveillance for SARS-CoV-2 detection and quantification. I've been working for 3 decades in sewers, and we thought to harness this experience to combat the SARS-CoV-2 pandemic. So as you can see, as you can see on the slide, patients or people who are diagnosed with, or even not diagnosed with COVID-19, they excrete. The virus itself finds its way to the wastewater either by washing hands or washing themselves, but much higher concentration is excreted by feces, by people who are positive, not necessarily diagnosed. And our idea is to look at the virus RNA in the sewers instead of looking at the virus on diagnostics.
Eran, you need anything?
Eran, you are mute. Unmute yourself.
Can you press enter, please?
Pardon?
Can you press enter on the slide?
Yeah.
All right, so what we're talking about is we're looking at populations, not on individuals. Since we're looking at populations, we don't need diagnostics for individual persons. We look at the whole population. We use the same method at the end, RNA qPCR. Luckily enough, the virus is not active anymore In wastewater, I won't go into details on that, but the remains of the RNA are there, and we're looking at the remains of the RNA. We don't need any consent from the people. We know that some people are not very happy to get diagnosed. We don't have to go to the people. And in a smart way, we can basically know what's the situation in a large number of people. Basically, if implemented, it's an early warning system. We can work on a spatial basis, and then we can spot hotspots in cities and urban areas, and thus give idea to decision makers. You don't have to close to quarantine a whole city, but you can quarantine a quarter or a neighborhood. And also we look at temporal changes. That's great. This is a representation of an as-made sewer system in a neighborhood in Netanya. This neighborhood, Netanya is a coastal city in Israel. This neighborhood is about 15,000 people. And you can see this is how the network looks like. It's a very hierarchic, tree-like network. Please, next please. Now, if you choose the right choice, if you make the right choice, you make the right choice and you then decide where to sample. If we sample where the sewer in a manhole around where it's written outlet pipe, then we get an idea of what's going on in the whole neighborhood. We can sample now 15,000 people altogether. Next, please. Now, if we look at the first branch going up, we can then isolate a small neighborhood of about 4,000 people, and we get them all at once, all right? Next, please. The same goes on. Next, please. Next, please. And another next. All right. So basically, no. Go back one. So basically, we can go upstream. We go from the outlet, let's say from the municipal wastewater treatment plant. If we get a signal there, then we go and at selected junctions, we put our equipment, and then we can go upstream and actually signal out where are the hotspots, where are the infected people concentrated. Next week, please. This is a large pilot study we have done for a week long in Ashkelon. Ashkelon is a city in southern coastal area of Israel, about 150,000 people. For Israel, it's not a small city. For Brazil, it's probably nothing. And what we can see, I won't go into details, but we get a heat map. All right, red is high infectivity ratio, yellow is low, and this is divided by neighborhoods as dictated by the sewer network. So we can see here in 17th of May, we had some orange neighborhoods, but still some yellow ones. And on the 25th, everything turned orange, and some neighborhood turned red, which means that the concentration in the sewers were higher. We can then convert it to number of people, and then the decision makers can make a decision. And surprisingly enough, we knew it before the Ministry of Health knew what's going on. Just to remind you that about only 10% of the people who are infected are diagnosed. So we sit on an iceberg, and usually the Ministry of Health and Health authorities see only the tip of the iceberg, and we are able to see the whole iceberg. This is the idea, a lot of work, a lot of difficulties in diagnosing and extracting and getting the samples and so on, but it's going on. We had a very successful pilot, and now we're working with Ministry of Health to have another one to refine the method. That's it.
Thank you. Now
Thank you very much.
I'll stop sharing, and we'll go to Mauricio Fares. Please, take on.
Hi, good afternoon. Can you hear me?
Yes.
Great. I will start sharing my screen. There you are. Okay, well, good afternoon again. Thank you very much for the invitation. My name is Mauricio Fares. I'm the CEO and co-founder of Untitled, and we are one of the companies incubated by Eretz.Bio. In Untitled, our vision is to empower the healthcare professionals with artificial intelligence solutions so we can improve our work conditions, improve our accuracy, and at the end, improve the patient experience in the healthcare system. Untitled Peak is our artificial intelligence tool aimed for radiologists that is currently ANVISA and ANMAT approved. Those are the regulatory agencies of Brazil and Argentina as well. And we are currently operating and working in the majority of countries from the Latin American region. So what we do is, we offer artificial intelligence services for many of the diagnostic imaging procedures. This is an example of a chest X-ray of a patient with abnormal condition within their lungs. Our system in less than a minute can tell if that chest X-ray is normal or abnormal, so the doctors can organize their workflow. And also we give diagnostic support for those that are not that experienced about several conditions, including cardiomegaly, opacity, pleural effusion, a series of conditions that are typically seen on these kind of images. And then we were all hit by this pandemic, COVID-19. And since we were an artificial intelligence company already working with chest X-rays, we decided we had something to do about it. Just a quick refresh of the diagnosis. We all know that the COVID-19 is diagnosed through PCR. That's the diagnostic gold standard, and it has a modest sensitivity. But then, in particular in our region, we have some issues with the test availability and the delay that we have obtaining the results. In our area, for example, here in Argentina, you can be waiting up to 6-7 days to get your result. And then medical images, just as chest X-rays and computer tomography, can be used as a support, in particular if PCR is not available. Can be used for management decisions, can be used to deal with complications of the disease, and to figure out if there are some alternative diagnoses that you have to make. So we asked the question, what if we use our system that it's been already trained by hundreds of thousands of images, and we repurpose it so we can be used to detect COVID-19 patients. And this is just an example. Here on your left, you see a patient with COVID-19, and the system is detecting the areas where the patient is affected by COVID-19. And this is a comparison with other types of pneumonia in panel B. And then when we calculate the difference between other pneumonias and COVID pneumonias, we can see that our system was correctly learning and separating the typical pattern of affection in chest X-rays. So the next question that we ask is, okay, will this be useful in clinical practice? So we conducted a multicentric study in Latin America with doctors from Argentina, Brazil, and Colombia. And we give them the randomized support of our tool. And what we found is that this was done at the very beginning of the pandemic. So obviously doctors weren't that trained to detect the disease, and they have a limited sensitivity and specificity. But most importantly, when we gave them the tool, most of the doctors have an improved sensitivity and specificity, and they work better detecting patients with COVID-19. And I think that was the main finding that we have with our tool. And that was one of the reasons why we made it publicly available for free to all the region. So as a conclusion, once again, I think COVID-19 gave us the opportunity to prove that the combination of radiologists or doctors with artificial intelligence is better and outperforms doctors alone. And the future is in this combination, that artificial intelligence for COVID-19 can be used as a support tool, obviously not as a diagnostic, that's for the PCR. And we definitely think that more research and more collaboration are needed to validate and support the further use of these tools. This work was done from Untitled and with collaboration from many centers. In our team was led by Francisco Dora and Marcel Serra. I think that's all. And the tool is available on that website, and I can you the link later. Thank you very much.
Thank you, Mauricio. Now we'll turn to Joachim Behar. And please, Professor Behar, Dr. Behar, you can share your screen. Are you muted? We can't hear you, Joachim. You're muted. Joachim, please unmute yourself. You're still muted. Oh, okay. Now you should be okay. And
yes, can you
please
hear
share
me?
the screen again.
Thank you. I will share screen. All right. So hi, everyone. First of all, it's a pleasure to join this meeting with Einstein Hospital. I was actually very close to the hospital a few years ago when I visited Sao Paulo for collaboration with the UNIFESP and the sleep clinic there. Briefly, my lab is focused on the use of artificial intelligence or machine learning for within the context of physiological time series. It's a year and a half old now at the Technion. And within the context of the coronavirus, we decided to focus our effort on looking at the oximetry time series, meaning the oxygen saturation measurement typically done using a pulse oximeter that is worn on the fingertip. Why is this signal in particular? Because COVID-19 is often results in the development of pneumonia. So we hypothesize that this lung impairment will eventually be reflected on the oximetry time series, which we can use for diagnosis or monitoring. So if we look at a vital sign, oximetry or heart rate, blood pressure over time, we can make a single point measurement, which is often what's done, or repetition of single point measurements, say every day at 2 p.m. you will take your heart rate. But obviously that is limiting and hiding the actual dynamic of your variable. So we focus in the lab on continuous monitoring of physiological time series and here oximetry analysis. So we can measure long recording of oximetry, for example, during night time. And you see here an example of a typical oximetry level around 95 of a healthy patient overnight, which is constant or usually about constant. Here, an example of a patient with COPD, where you can see a long desaturation event, hypoxic event, characteristic of COPD. Here, an example of repeated shorter desaturation, which is characteristic of obstructive sleep apnea, a sleep disorder breathing. And in this last example here, the accumulation of both COPD and OSA and sleep apnea, where you can see this long hypoxic event and embedded within it an obstructive apnea event. What this reflects or this aim to highlight is that there are patterns or a certain dynamic in the time series of oximetry that allows us to diagnose or monitor respiratory disorders. And so in the case of COVID, this is a way to diagnose or monitor patients with the coronavirus. We have developed a software associated with this work, which implements all the oximetry digital biomarkers we implemented. So we implemented a new library in Python and an associated software that goes with it, where you can see here the oximetry time series and some pattern highlighted that are detected, our digital biomarkers. And this is something that will be in preprint in the next few days. Where are we with this project? Basically, we developed all the tools we need, and we proved on COPD and those are in the lab that this is working. And we are talking with a few potential medical partners to get access to COVID-19 oximetry data, whether of patients that are recorded remotely with a mild condition of COVID, or in the intensive care unit where they are continuously monitored. And of course, we are, I would welcome a collaboration with the Einstein Hospital if there are MDs interested in looking at this topic. We have all the tools to do that analysis. I will finish by thanking Jeremy Levy, my student who I'm working with on this project, and thank you for your attention.
Thank you. And the next speaker is Caetano. Please take the command.
Hello, folks. Can you listen to me?
Yes.
Just give me a second to share my screen. All right, it's loading up. Okay, just give me a little second. All right, guys, so I'm going to talk to you about a subject that is being very much talked about throughout the entire world, which is ultraviolet germicidal radiation for elimination of the new coronavirus and also many other microorganisms. So what is this ultraviolet light, right? It's just some sort of electromagnetic radiation, just as radio waves, microwaves, infrared radiation, but it has a little more energy. Its energy range is actually in between visible light and X-rays, but ultraviolet light is a little more selective. It actually interacts very well with DNA and RNA. So you can see here in this graph that the RNA curve is this one on the left, the DNA curve is the one on the right, because they are shown due to their uracil and thymine composition. And right now we have two major technologies that can be used for artificial decontamination of surfaces, air, and even water using ultraviolet light. The latest one is LED technology. However, I must say that this technology is not very mature yet. The efficiency of those LEDs is not as great as those for the classic quartz tube lamps. And you can see here as well that their emission wavelength is not very well matched with RNA, which is the genetic material of the new coronavirus. So ultraviolet light acts directly upon DNA and RNA damage. It causes the formation of pyrimidine dimers that inhibits the translation of DNA or the replication of DNA and the translation of RNA. So due to that, any microbe is sensitive to ultraviolet light. Doesn't matter to me if it's a virus, bacteria, fungus, or parasite. If it has DNA or RNA, it is susceptible to ultraviolet light. And it's not just magic, right? It's not just shining the light upon some surface that you're going to have some effective response. There is a dosimetry linked to ultraviolet radiation, which is, in simple terms, it's the power density of the light. It is the power being irradiated of a specific area, and it's multiplied by the exposure time to know the total amount of light that we deliver onto that surface or to that volume. Okay, and then we have ultraviolet dose. And then we can have some more analysis. For example, we're the first company here in Brazil to prove the effectiveness of ultraviolet C light against the new coronavirus, the SARS-CoV-2 strain. And we can see here in this table and also in the graph, it's a very fast method. You can see that within less than 0.01 second, we can already have 90% of fire inhibition. And after 0.3 seconds, we already have 99% of inhibition. So that's very effective. And oops, there you go. And now we know that the coronavirus can live outside of the human body for days. If it's on plastic surfaces, it can last for 3 days. If it's just floating on the air, it can last for up to 3 hours according to this study, but there are other studies showing that it can live for 16 hours. And then we developed some specific weapon for this use of these pandemics. This is the portable handheld irradiator for surfaces. You can use that for inactivation of the virus in several types of surfaces, including the most complicated ones, such as electronics, papers, tissues, fabrics, even food, but also for simpler ones such as glass and plastics and steel and so on. This is an equipment that is very interesting for this time now. It's a ultraviolet equipment for reprocessing face masks, and we proved the effectiveness of it for N95 and also for cotton masks. It gives a more than 99.999% of microbial inactivation using this method. And we also proved that after 60 cycles of decontamination, the N95 masks perform just as new. We also have equipment for air decontamination. This is a very messy situation that we put our equipment to test. This environment here is a collective dental school. So they do all their dental practices using this equipment here. And we showed that we can give more than 99.9% of microbial elimination from the air in this very challenging situation. We also have some new equipment coming on. We're developing in partnership with the Einstein Hospital, these equipments for entire room disinfection. This should come out within a few months from now. We also have this new equipment that should be issued on next month. That is replacement on the roof of a room. You can be either this linear model or this corn-shaped model that is easier to install. They all have lots of presence sensors that are redundant, like we have infrared sensors and also microwave sensors. So it's very safe, and we're here to illuminate hospitals and fight COVID, to bring healthier environments for people to work and for patients to be in. And this is it. I'm Caetano Sabino, I'm founder of Biolambda, and thank you very much for listening.
Thank you. The next speaker is Professor Noam Gavrieli. Noam, please take the stage and let me know when you want to flip the video or you want to do it by yourself. Go ahead.
Okay, thank you so much. I will let you know it will be towards the end. So I'm in Chicago. I got here 3 days ago. The only way in my advanced age that I could do so was by protecting myself with the Viri mask. Now, the Viri mask is this. I was wearing it all the way, even watched movies and slept with it. The Viri mask has a frame and it has a filter. The filter can be detached, and as you can see, it has HEPA material inside it, which is designed to block viruses. Unfortunately, the material used in these masks and also N95 allow particles that are under 0.3 microns to penetrate. The coronavirus is 0.12 microns. So it can penetrate. It also does not seal around and does not seal around the eyes. So we started this project exactly 6 months ago when we realized that the COVID-19 is an important pandemic, and we understood that the penetration is through the respiratory system, including the lining of the eye, the conjunctiva. The COVID penetrates the respiratory cells, multiply in them, and then after enough copies are made, it becomes symptomatic or asymptomatic disease and is detectable. So we decided to try to create a totally isolated environment around the respiratory system. This meant having a seal all around the face, eyes, and face, which is similar to what we learned from gas masks in the early 1990s and again in the early 2000s. We decided to make a filter that can actually block the viruses and not only bacteria. And we wanted it to be comfortable, meaning it is not difficult to breathe through it. All of you who have tried the N95 know how difficult it is to pass air in and out. So our understanding of the respiratory system allowed us to design a product that can block the viruses, meaning that it is very tight mesh or sieve, yet not create a high resistance to breathing. We started the project in a platform that we have been running for years. Actually, the products, medical products in orthopedics, are being sold in Brazil, including Einstein Hospital. We used that platform in order to very quickly develop the concept, design it, order the needed molds, and start production. We started shipping products about a month and a half ago, and it is now being sold and ordered all around the world. This is pretty much an engineering project that is aimed and designed to target specific requirements. We identified the need and worked out the specifications from bottom up. Rafi, can you show the video? Just to give a little bit of a feel of where it is used. So the product is designed to be used by medical personnel who are exposed to a high risk of infection, but also to be able to isolate every one of us, and by doing so, to be able to jumpstart economy, jumpstart cultural events. Only today I was asked whether it can be used in theaters and concerts. So the answer is, of course, yes. In international flights, in schools, in banks, by police people. Essentially, everyone can use it, and everyone can be isolated, as well as isolation can be probably even better than isolation in a house or a room. Go ahead. It's a very short video from Nahariya Hospital in Israel.
Did you see it?
No, it did not run. You need to double click on it.
Yeah, the problem. No, the problem is it runs immediately. So here, let's see.
Let me show.
Okay,
Are
do you
you
see it
share?
now? Do you see it now?
Are you share, Rafi?
Yeah, I share and I run it, but
One moment, please.
So you try. I'll unshare and you share. Let's see if it works.
Okay, this is a surgeon in the maxillofacial department at Nahariya Hospital. These doctors are exposed to large amounts of aerosols, so he's using the VRE mask. So I wish to thank the organizers and all the participants for this opportunity. It is lovely. Thank you so much.
Thank you. And now we turn to Anselmo. Please take over.
Hello, can you hear me?
Yes.
Oh, okay, great. Just a minute. Please let me know if you can see my screen.
Yes, we can.
Okay, thank you. Thank you for
make it presentation mode, namely that it will take the whole screen.
Okay, just a minute. It's just sorry. Is the read mode here? I don't know if that works. Here we go. Okay, thank you for inviting us to this call. We really appreciate the invitation to participate to this webinar, and we are really honored to be here. Let me introduce a little bit to Casper Biotech. Casper Biotech is a biotech startup founded in Argentina and now based in San Francisco in the US. Our mission is to create a new standard in molecular diagnostics, and for that, we are leveraging the power of the CRISPR technology. The CRISPR technology has been known for many years as a powerful tool for genome editing or gene editing, but CRISPR itself, its core functionality is to work as a machine to identify DNA or RNA sequences. So CRISPR itself, by doing that, is also a powerful tool that can be used in molecular diagnostics for any other purposes. It's a platform that could help, for example, to detect infectious diseases, to detect microbial resistance, and so on. So from the very beginning, Casper has been working in the development of many solutions in the molecular diagnostics field. Especially at the very beginning, we started working in the development of tests for detection and diagnosis of tropical viruses like dengue, Zika. But of course, this year, due to the pandemic situation, we really focused all of our efforts and all of our resources to the development of a new solution for the diagnostics of COVID-19. And the main question on the main need that we found was that in the current scenario, molecular diagnostic is mainly limited to high complexity settings or to highly trained professionals. So this is how, I mean, this is where Casper makes a difference because we are developing solutions that are applicable, that are instrument free or require very low capital expenditure and are very applicable to situations or to low resource settings or low complexity settings. In a few words, we are working on two solutions. One of the solutions is what we call the Lyo CRISPR kit. And this kit has been already fully developed. It's a kit that is applicable to, as I mentioned, to low complexity settings, to low resource settings, because it requires just, as we will see further along, it requires just a few instrumental, and it's very easy to, it has a very user-friendly workflow. So all the reagents are lyophilized, so that requires minimal operator intervention, reduces the potential cross-contamination. This kit has been already submitted to FDA for approval. So we already had a very good feedback from them, and we are waiting for the approval possibly in the next 2 to 3 weeks, and this kit will be available in the market. And then we are also working, that solution, the Lyo CRISPR kit, meets a demand on those kind of settings for decentralized labs, for example, hospitals, or maybe shopping, an airport. But then we also are working on a solution that is a truly point of care and a fully automated solution. We call it the phantom point of care. The phantom point of care is, it's been a design that has a hard work from the engineering team. It's the size of a smartphone. It requires just for the patient to get a nasal swab and introduce the nasal swab into this device here, into this second image we have here. You have a device and you have a cartridge. The cartridge is the part of this device or the part of the solution that is disposable. You just have to place the swab, the patient swab, into this device, and you get results in only 15 or 20 minutes. This solution is at this moment being developed, is being validated, and we are planning to submit this to FDA approval by mid-October and hopefully have the approval by November or December. This is how the workflow for our kit, for our Lyo CRISPR kit, and how our biology works. It requires to collect the sample. We designed a kit that works not only from extracted RNA, because we know that RNA purification kits, the demand is really high. So we designed also a solution that works directly from sample without any requirement for prior RNA purification. After the sample is collected, you just need to place the reaction into, incubate the reaction at 60 degrees. So it requires only a heat block or an incubator. That's all the instrument it requires. And then the last step is the detection by CRISPR and the fluorescence readout in a affordable and portable device that we also designed, that is called the fan box. This box that we call the fan box, you have to place the reaction from step 2 into this box, and the results are automatically interpreted, and the patient results are easily visualized in a software application connected to a computer. So this is data from our validations, our clinical validations that we perform, and then we submitted to FDA. We had several partners in the world, health institutions that help us to conduct these validations. With and we really met high standards of specificity and sensitivity, and a workflow time of approximately 60 minutes to process 96 samples in parallel. So this is basically all we have to share with you. And thank you very much again for inviting us for this webinar.
Thank you. The next speaker is Professor Avi Schroeder. The problem is he is with his family in a place where he doesn't have a good network connection. So I'm trying to see if I could share the video that he recorded for us in order to share it. Can you see it now so I can play it?
Rafi, I can share for you if you want.
Is it shared now or not?
No.
No, okay, so please share it.
Okay.
Very good. So let's turn on the volume and we'll go.
Hi, everyone. My name is Avi Schroeder. I'm an associate professor of chemical engineering at the Technion-Israel Institute of Technology. My group studies the connection between nanotechnology and medicine. And thank you very much for joining this seminar today. So when I entered the field of nanotechnology and medicine, what inspired me is these three images in front of us. They come out of a clinical paper that showed that nanoparticles injected intravenously, so directly into the vein, can actually target different types of tumors inside the body. They can detect the diseased tissue where they will localize and then release their medicines locally. This, of course, is very important for cancer, as we can see here with 3 patients, totally different tumors, one at the base of a tumor at the base of the tongue, a patient with breast cancer and metastasis in the local lymph node, and on the right-hand side, a patient with lung cancer and metastasis to the spine and the bone marrow. And we can just imagine, for treating coronavirus, having a medicine that will be injected intravenously and target the lungs, where it will release a medicine that will change or reduce the viral load or prevents the cytokine storm. That can have huge impact on the patients. So nanotechnology is about delivering sophisticated medicines in a very elegant manner and a targeted manner to the disease site. And when we look at the nanotechnology, the way I always imagine the nanoparticles are small tennis balls, the size about 1/1000th the width of a hair on our head. So a nanoparticle actually is smaller than any cell we have inside the body, but can contain a payload of medication that can treat a single cell or even a cluster of diseased cells. The nanoparticles require a lot of engineering when you come to develop them. For example, the liposomes we have in front of us, these 100 nanometer particles are composed of about 100,000 molecules that contain inside them also a medicine that is protected in the blood but is released in a therapeutic dose at the disease site. So these particles have to have the ability to circulate in the body and detect the disease site where they'll release the medicine in a targeted form. And just to emphasize how small nanotechnology is, because 1/1000th the width of a hair is something which is hard to imagine, but this is another example of what nanotechnology is about. So you can see here a silicon chip on the tip of a person's finger. This was taken at the Technion, and actually this small silicon chip contains on it the entire Bible in Hebrew, which is engraved inside the silicon chip, including the small notations that Hebrew letters have that are the vowels of the Hebrew language. So nanotechnology really contains a lot of, in this case, information in a very, very small manner, or in our case, a lot of medicine in a very small particle. And this alone, the ability to target a disease site, has had huge impact on the clinic. For example, Doxil, which is the first nanotechnology to be approved by the United States Food and Drug Administration, was invented in the Hebrew University in Jerusalem. It's called Doxil, and it contains 100 nanometer particles, liposomes, with an anti-cancer medicine inside of them. In this case, the drug was approved in order to reduce side effects the chemotherapy had on patients. And once it was loaded into the nanoparticles, it released primarily at the disease site, so it had very few side effects on the patients. And it, of course, also improved the therapeutic efficacy. But nanoparticles can also be administered by inhalation. They can contain multiple medications inside of them, so you can treat different aspects of the disease or different disease pathways using nanotechnology. And you can also deliver very sophisticated medications, such as RNA-based medicines, which can either shut down the production of proteins inside the body or turn on the production of therapeutic proteins inside the body. One very famous example is the recent clinical trial Moderna is running with the nanoparticles that, or specifically lipid-based nanoparticles that contain inside them RNA molecules, messenger RNA molecules, for encoding for proteins that are related to coronavirus, and by that vaccinating patients against coronavirus. So nanotechnology is playing a very important role, not only in treating diseases such as cancer, infection, and inflammations, but also treating coronavirus infections too. And this is just one very extreme and very important example. We too in our lab are working on using nanotechnology for reducing the viral load, specifically reducing the amount of virus inside the infected lungs and targeting our nanoparticles to the lungs of the patients. Our researchers are working day and night, literally, to create these nanoparticles that, one, target the lungs, and two, reduce the viral load, reduce the amount of viruses inside the patient's lungs, in order to prevent the cytokine storm that we see in many of the patients, and also to improve their therapeutic outcome. So in the 5 minutes I have, that's a very short emphasis that nanotechnology is playing an extremely important role in treating coronavirus, with a lot of innovation inside, but especially a bright future for overcoming coronavirus using nanotechnology. I'll be very happy to entertain, try to answer questions people may have, primarily by email. Thank you very, very much.
Thank you, Professor Schroeder. Now we move to Gustavo. Please take the command.
Hello, good afternoon and good evening. I share my screen. So thank you for Einstein team for the invitation. So my name is Gustavo Simões. I am co-founder and Nanox CEO. So Nanox is the first Brazilian technology company. We started the company in 2006, and we using bottom-up and nanotechnology process to create sometimes nanoparticles, sometimes microparticles, building the molecules using some materials. We are a corporation, and we have some investors to invest money in the company to grow the business. So basically, Nanox makes some silver-based materials. We create different kind of technology to use 10 or 15 times less silver than our competitors around the world. So we have patent in Brazil, Europe, and the US. Our product is approved to FDA in United States. We are applying to EPA. We approve in Anvisa in Brazil, and we are compliant with Europe regulation. The product is no toxic. Basically, we create around 20-25 different products. Can be powders, can be master batch or plastic with powders, can be liquid. But these products, in the end of the day, Nanox has antimicrobial performance to kill bacteria, fungus, and virus. Depends on the market. So we sell for packaging, for food packaging, for wood laminates, for plastic, for different utensils, for home appliances. Our products can be applied in different kinds of materials and in different markets. During the pandemic, Nanox can prove the first finishing process to fabric and the textile. We prove our product kill COVID-19 in 2 minutes of the contacts. Our product can be applying, for example, in textile or fabric, in any kind of fabric, synthetic or organic, in cotton, in PP, in nylon, in PET. It's possible to apply and to build any kind of fabric, like clothes, like mask. But our innovation here is to show the first textile to kill COVID-19 in 2 minutes. We are working together with São Paulo University in Brazil. São Paulo University isolates the COVID-19 in Brazil in the beginning of the February. And we proved this using the ISO 18184. Is a virucide standard, and we add some virus, and after 2 minutes we measure of the reduction, and it's possible to show the silver materials can eliminate 99% or 99% in 2 minutes of the contact. This product can be applying in any kind of fabric or textile and can make different products. So basically, another interesting case, Elka is a Nanox customer to make toys. In the beginning of the pandemic, to start a project to make some N95 reusable. This is the first plastic reusable mask. In this case, we are applying our antimicrobial antivirus on the mask, and the mask is reusable. So for the hospital decrease a lot of the cost of the investment in mask, because in general N95 is disposable. In this case, only the filters are disposable, and the mask is, you can use around 3 or 4 months, depends on the application. The product has Anvisa in Brazil. We make the standard test to prove the N5 efficiency. So this is a interesting case in Brazil. So another is one film, PromoSafe makes some protective films. These films has our product, our antimicrobial. We make the protective film in different size, and this you can apply indoors and in the public areas, especially in some equipments on the hospital, and the product can protect and kill the bacteria and fungus and virus in 3 minutes of the contact. So is the technology is the same almost the same of the textile on the mask and the film. The difference sometimes is the silver concentration, is how can silver are available on the surface, because if the virus has contact with the silver, there is it's like oxidation in the surface, and this oxidation broke the virus, and the virus, especially COVID-19, kill in 2 minutes. So the next launch maybe will be the one spray. It's like this spray. One spray you can spray in some rooms and some different parts of the hospital, of the home, and this is we will be available for the next month using the same silver-based technology. So thank you very much. This is my website. If you have some question, we'll be here. Thank you.
Thank you very much. The next speaker is Professor Yoav Shechtman. Yoav, please take command, and I guess you're going to share your screen. Please.
Yes.
Okay, go ahead.
Okay, do you see? Oops.
Do it in presentation mode.
Okay, you see it?
Yes,
okay, thanks. So thank you very much. First of all, thank the organizers for inviting me to this really interesting webinar. And boa tarde to the Brazilians. I would like to present our microscopy method that will hopefully enable antibody detection at very low concentrations. So my lab develops novel optical microscopy methods to observe life on the nanoscale. When the whole corona threat became evident, we asked ourselves, what can we do to help? And unfortunately, we don't know anything about vaccines or therapies, but we do know how to image single molecules. Here's an example of a routine measurement that we do in my lab, which is single molecule imaging. What you see here, each one of these shapes is a single fluorescent molecule. And the shapes appear different because of special optics that we develop, optical components that we develop in the lab. And these encode the depth of each molecule. So from such a movie that we capture, we analyze it using our software, and we end up with a 3-dimensional image at super resolution. This is mitochondria in mammalian cell at sub 50 nanometer resolution. So this is the kind of things that we do. Now, if you have this technology, you don't have to create a 3D image of something. You can simply track molecules that move. And so this is the basis of our idea for diagnostics. So our approach is as follows. It's in early stages, but let me lay out the plan and show you some preliminary results. We start with a patient sample. In the future, we will. We label the entire serum. This is for antibody detection. So with a kit that fluorescently labels all antibodies, whether corona or others, we mix it with labeled antigen. This can be the spike proteins, in our case, of the coronavirus, or a part of the spike protein. So now we have two colors. We have red and green of two different species. Then we mix them, we flow them in a flow channel, and we image it using our microscopy. Now, whenever you see a co-localization, it tells you that something bound is bound together. So it means if you had a green and a red spot moving together, then it suggests that there is antibody in the solution. Otherwise, the antibodies won't bind specifically to the spike proteins. And so we take this, the idea is to take this signal, to analyze it, and seek these co-localizations. That is the strategy. It's pretty simple. Let me show you what this looks like. So what you see here is strange shapes. And let me tell you what we are looking at. So we have in red red labeled spike protein and green labeled anti-spike antibodies. And we know by looking at these shapes that this is a red guy, this is a red spot, and this is a green spot, and we know where they are in 3 dimensions. So I can also tell you that this green spot is exactly at the same 3D position as this red spot. They appear shifted because of our optics, but they're at the same place. And the same is true for this pair, for the other pair. Now, if it just stands there, it's hard to say, but then we start flowing them, and you see these guys are moving together. This pair is moving together. This means they are bound. This suggests that in this sample, there are antibodies to the spike protein. So what we have here is the potentially detection technique for detection at the single antibody level. Very, very sensitive, which could be in principle orders of magnitude more sensitive than traditional state-of-the-art high-end approaches like ELISA. So where are we now? For COVID-19, of course, the technique can potentially enable very high sensitivity detection of antibodies. Also virions, I didn't talk about it, but it's a similar idea. If you're going to start pooling in tests and lower the concentrations of antibodies, it can be important. But I have to say, I know the theme of this webinar is COVID-19, but the application of this technique can in principle be more general. We are currently discussing with doctors, physicians looking for detection of other biomarkers that appear at low concentrations where this can be very helpful, this technique. Currently, we are at method validation process. We have a patent submitted, a grant application. Finally, I would like to thank the guys for my group who did the work, the members of the group and the entire group, and you for listening. Thank you very much.
Thank you, Yoav. And the next speaker is David Emmanuel from Brazil, please.
We'll share my screen. Can you guys see my screen?
Yes. Go ahead.
Okay. So thank you for the invitation. I'm really glad to be here. I'm going to talk about massive molecular diagnosis for COVID-19 using NGS data. And first, I would like to talk about the VirStation, which is a startup that was created inside the Albert Einstein Hospital. It's a branch of the hospital. It's a branch that was born inside the clinical laboratory, and it was thought to be a cloud-based solution for bioinformatic analysis and to support interpretation of genetic tests for NGS data. So by genetic tests, I mean exomes, gene panels for cancer, for hereditary disease, and all this kind of data, sequence the genetic material of the patient and provide support to geneticists, to physicians to analyze this data and be able to provide a meaningful diagnosis of the condition that's being studied, it's being investigated. So, so we had a target problem because we are inside of the laboratory of the hospital, Albert Einstein laboratory. At the very beginning of the pandemics, we were able to see that it was clear that there was a shortage of molecular biology supplies related to RT-PCR. So our laboratory, we have prepared ourselves at the very beginning of the pandemics. We have created the RT-PCR assays to be able to diagnose COVID-19. As a matter of fact, we were the first laboratory in Brazil to detect the first case of COVID-19. So at the very beginning, we start to see that there was a shortage of molecular biology supplies, not only in Brazil, but in the entire world. And in Brazil, it was a little bit more more pronounced because Brazil do not significantly manufacture these molecular biology supply. Especially, I'm talking about the supplies for RNA extraction. So this was a very important limitation that was that make us not able to scale testing to take this to another level, take the testing for another level. And also we thought we've seen the difficulty to scale testing because of the equipment limitations. So the real-time PCRs equipment, so we would have to buy a lot of those equipment to be able to scale testing. And we also would see a sample handling problem and also results rendering problem. So here I brought to you a news that was published in the New York Times. It was July 23rd, like 2 weeks ago. And the laboratories in the entire world, they are still seeing this short of supplies, this difficulty to scale testing, to scale the molecular testing for COVID-19. And this was the target problem that we've identified and that we tried to address. And we created a solution. We came up with the solution that we call VARS-V, which is an NGS-based molecular test to identify SARS-CoV-2 genome fragment. So it's very important here that it is a test, it's the exam that is able to identify a fragment of the genome. So it's a molecular test. We know that there are different tests, the serological test for identifying immunoglobulin ones. But with this molecular test, we are able to identify the virus at the very beginning of the symptoms, and we've been able to create this test. We've been able to come up with a solution that use different supplies from the RT-PCR. Especially, we use a 10-minute step RNA extraction. We use supplies that are very abundant here in Brazil, so we managed to address that concern. And also we are using to this test NGS supplies. I don't know how familiar you guys are, but NGS, we call NGS when you use sequence the genetic material. It may be RNA or DNA. When you sequence that material to analyze it in a bioinformatics, in a computational way, to provide results. So there are machines that do this kind of sequencing, and so there are a totally different kind of supplies that are not using RT-PCR. And we also being able to create a complete workflow to address this problem. So also the problem for sample registering to support wet lab work, and also the bioinformatic analysis of the data that is generated by the sequencers. We also, all of this is inside this solution that we create, which is the VARS-V test. So just to give you guys an example here, it's in Portuguese, and I apologize for that, but just to give you an idea of how our test works, you have first the patient sample, you collect that sample. We extract the genetic material with this very quick step and very simple step. And also we have kind of PCR to amplify some regions of the SARS-CoV-2 genome. And then we sequence that on the machines, the sequence machines, and analyze those data in our platform, the VARStation platform. So we've created this solution, and now we can see how it's what is the impact that it's having on the pandemic. So with this test, we are able to multiplex and to provide results, individual results, for up to 6,144 samples in just one run. So we've been able to scale tests scale on the molecular test in a very significant way. We also have been able to decrease the price of the RT-PCR, because compared with RT-PCR test, it costs about half of the price. So we also have been able to accomplish that. And all of this with an accuracy of 96% compared with RT-PCR, which is the gold standard. A lot of you guys have talked about that. So here's how our dashboard, like our platform, it displayed the results. It was a run for 1,100 samples, and we have the detected results, the not detected, and also the repetitions. And I'm not being able to tell you guys about the validation process that we had done, but we have published a preliminary result in the Meta Archive when we are exploring and showing the results of the validation process, showing the accuracy, how we compare with the RT-PCR samples, and the results that we have obtained. So thank you.
Thank you, David. And we'll move to Professor Joshua Schnittman. Please take control.
Thank you very much. Hi, good evening, good afternoon, everybody. I'm joining in from Switzerland. I'm an associate professor at the Technion in the Department of Biomedical Engineering. There's a lot of familiar faces here. It's an absolute pleasure. Thank you, Rafi. Thank you, Shlomo. I'm so happy actually to see Noam. Noam knows what my business is all about. He's followed my career for the past 10 years. As many of you probably know, the leading cause of COVID-19 related deaths is one form or another of ARDS, acute respiratory distress syndrome. Call it what you want. It fits somehow under this umbrella with basically mortality rates that hover around 40%. If you get on a ventilator and you're anesthetized, there are to date no pharmacological treatments available. This is not new. This is the state of the art for 40 years. Basically, all sorts of things have been tried. The thing is, the twist is that ARDS has been and was until 2020 an orphan disease, and suddenly it took really center stage. My lab has been a lab that deals, not unlike Noam's group, was in basically lung physiology, drug delivery. And we were working on ARDS, and sort of by a twist of fate, what we were working on suddenly had a whole different level of relevance, which is, can you think of some kind of rescue therapy in ARDS? This is not to say it is a cure to SARS-CoV-2, but once you experience a form of whole lung failure, if you don't deal with that, there's nothing that's going to get you out. If your lungs don't work, there's no oxygenation, you will not survive. That's the end of the story. Now, one of the peculiarities of ARDS is it has a kind of a sister or brother disease that affects infants, which is called RDS these days. It fits under ARDS. It used to be called infant respiratory distress syndrome. One of the characteristics that it shares, now it is a whole different beast, but it shares one thing, and one thing that the SARS-CoV-2 deals with, which is destructing the cells that create surfactant. Now, there is no human life or other mammalian life without surfactant inside our lungs. So it's been hypothesized that if you can rescue, if you could deliver basically surfactant to the lungs of ARDS adult patients, there's a chance to re-trigger this rescue of the lungs. And the crazy thing is that it works tremendously well in infants. And Noam knows this very well, and many of the MDs here across the planet know this. If you instill exogenous surfactant, you save neonatal lives. And we do it so well that it's a near 100% yield. But that technique does not translate easily to adults. And our group, and it boils down to one of my students, which actually pitched this idea 3-4 years ago to Noam at a conference, is instead of pushing liquid into the lungs, why not use a carrier as foam to defy the effects of pooling and gravity? In other words, there's a scale problem, okay? Our lungs are huge compared to that of a 1 kilogram neonate. And so one of the hypothesized mechanistic reasons behind why potentially this rescue therapy did not work in adults is that simply we cannot get what could potentially be a life-saving drug, okay, because we don't know how to distribute it inside the lungs of adults. And so over the course of the past 2 years, we've shown ex vivo and in vivo in preclinical rat models that we have safety and efficacy with a foam surfactant. And as I speak today, really, this is in the process, we're running the preclinical trials on adult full-size pig lungs, which are 60 to 90 kilograms, using this foam surfactant technique. Now, what our ex vivo inside these lungs showed is that actually you can yield extremely potent distribution inside the lungs, no matter the orientation of the patient, here be it the pigs. And so basically, to make a very long story short, I'm hoping in the next 2-3 months to come back to the table and show you that in models of ARDS that are induced through lung lavage, which was done in the rats and now in the pigs, you can basically bring back to baseline basic oxygenation and the classic metrics of quote unquote an operating lung. And that's essentially what's trying to be done inside the ICUs. So the technology, which is called liquid foam therapy, which was patented in my lab and is running through research in my lab, is now being licensed to a startup that came out of my lab that was founded, a month or two ago, which is called Nishima Medical. And its CEO is none other than the inventor and my former PhD student, Dr. Jan Ostrovsky. And so basically, if this works, and there are no promises, but if it does work, then there is reason to pass this into phase 1 human trials. So a lot is at stake here. And basically this technology is going to translate from my lab to the company as we move forward. And I'll leave it at that. Thank you very much.
Thank you. Our last speaker is Professor Ezri Tarazi. Ezri, please take command. You would like to present from your PC or would like us to? Please go ahead. Really unmute your PC.
Why can't, wait a minute. Okay, sure. Do you see it? Do you see the presentation?
Yes, we do. Go ahead.
Okay, so time is short. Our approach to the issue of protection, we call it air shield. The system is going to close the gap between the third level of protection with medical teams to the gas mask protection, which I was involved, and you see here one of the options using blowers that create active protection through filters. So we all know the problem in Israel, at least right now, 3,500 medical teams members got a corona. So our objective was to create something that will have first, better protect the teams from aerosols. 3, clearing the fog from the eyes, from the shields, and of course, create better comfortability, CO2 and other things that they face. So we have a system that based on a blower that we developed with a filter, HEPA filter, of course, a very light pipe, and the main invention is a manifold. So the first version is the version that is open. You see it here. The visor get another shield of air, clean air, that add protection and comfortability. And you see here our specific patent pending way of designing the way the air is coming out from this manifold. It's all creating a kind of a curtain of filtered air. And we 3D print it with our printers in our lab. It's a new HP 580 that can create from powder a very, very stiff and very good product. And it can manufacture 40 like this in one batch. And then, of course, we have another option, which is using a hood cover for a better protection and comfortability that gives 100% protection and also comfortability, because you can take off even the mask. You don't need the mask in this version. Overall, the idea is to better protect teams. We tested with several hospitals in Israel. You see here, Rambam Hospital, Carmel, Bnei Zion, and even one in the US, the Holy Name Medical Center. We also tested in the Hadassah hospital with those who create the aerosol, which is dentists. So thank you very much.
Thank you, Esri. This completes the list of the presenters, and I would like now to defer to Professor Shlomo Meital to conclude this session. Please go ahead.
Thank you, Rafi. So I have the honor of closing this webcast with some very brief remarks. The science fiction writer Arthur C. Clarke once said that truly advanced technology is indistinguishable from magic. Today, I believe we have seen a dozen truly magical creative ideas, each one focused on helping our ailing world to deal with this novel coronavirus that can only live and reproduce by invading human cells. The novel coronavirus has forced humanity to think deeply how to fight and defeat this terrible enemy. Just a few strands of RNA. Our doctors, engineers, and scientists have marshaled their creativity. When we defeat this virus, and we will, it'll be because of them, their inspiration, and their perspiration, working long hours in their labs in Sao Paulo at Albert Einstein Hospital and in Haifa at Technion. In the end, it's creativity that will defeat Corona. You know, Haifa is 10,666 kilometers from Sao Paulo. We hope this webcast has shortened that distance. We believe it will mark the start of a long and fruitful collaboration between Technion, where Startup Nation was born, and Albert Einstein Hospital's Innovation Center, the home of brilliant entrepreneurs. So let me extend some thanks. Special thanks to our dozen presenters. It was fascinating. To Clarice Fuchs of Technion Brazil, who organized this webcast and ran it perfectly. To Claudio Terra, head of the Einstein Innovation Center. To Dr. Kleiner and Professor Wolf, who introduced the webcast. My friend and colleague, Rafi Naveh. And most of all, thanks to the many participants in Brazil and in Israel. We'll do our best to respond to your chat questions offline. Toda Yisrael, obrigado, Brazil.
And
thank you.
Thank you. Thank you.
Rafi and Shlomo, thank you very much.
Thank you very much.
Thank you very much. This concludes the webcast, and we look forward to your remarks and your feedback offline, online, wherever. Have a great one, and especially good health to everybody.